So our project is on the effective distance from habitat fragmentation on biodiversity of invertebrates. I'm Coleman Taylor.
I'm Diego Cifuentes.
At the Department of Biology at Longwood University with Dr. Kenneth Fortino for Biology 341.
So let's start with the risk of habitat fragmentation. It basically comes from how it affects biodiversity. And how it affects the shift of its habitat. It is stated that due to fragmentation, biodiversity has a trend of reducing by 13% to 75%. Habitat fragmentation actually happens when human interaction causes an organism's environment to emerge in discontinuities in their habitat. This is mainly caused due to human interference, but it can also actually happen naturally.
So the research question for our experiment was essentially to see if distance was a factor when it came to fragmentation. So the further away the habitat is from the fragmentation, then the more abundance there will be. So like if it was closer to a fragmentation, there would be less abundance. The hypothesis for our experiment was that due to the distance between our sites and the trail or the fragmentation splitting site A and B, that the sites would be statistically different from each other when comparing diversity, richness, and then also abundance.
The methods we took to accomplish this experiment was first find a location, which ended up being the floodplains in Lancer Park. After we located a location that also had a man-made fragmentation, which we established to be in the bike trail in the floodplains, we then placed one site on the left side of the bike trail and then another site on the right side of the bike trail. And then a third site was actually placed on the left side of the bike trail at the same distance that the bike trail made from A to B is now on B to C. These sites had 5 pitfall traps on each of them, and the invertebrates that were captured inside the pitfall traps were then counted 3 times a week for 3 weeks. And then the invertebrates that were captured were then let go. The results shown in figure 1 demonstrates no relationship between the diversity and each site A, B, and C. Each dot shows the diversity for each day that the data was collected. The first figure shows no statistical difference between the diversity and each site with a p-value of 0.9209. In figure 2, this depicts no relationship between the total number of invertebrates found in each site compared to in each site. Each dot shows the amount of invertebrates found for each day that the data was collected. Now the second figure shows also no statistical difference between the total number of invertebrates of each site with a p-value of 0.2708. Now, the third figure illustrates no relationship between richness and each site. Each dot shows richness for each day that the data was collected. The third figure shows no statistical difference between the richness of each site with a p-value of 0.3232.
So just to conclude some of the stuff after the results section, our hypothesis is going to be rejected on the basis that our data was not statistically different when comparing the sites to one another, because there was not actually a p-value that was below 0.05. So none of the data could be considered statistically different from any other of the data points. It is possible that the distance between sites proved to be what led to each figure's inability to accurately display a statistical difference. So in the future, it could be possible that we or someone else could do another experiment with increased distance between each sites. I believe it was about 5 feet. It was 5 feet from site A to the trail and from the trail to site B, and then 10 feet between site B and site C, I believe. But it is possible that someone could do another experiment with increased distance between the sites to allow possibly for less overlap in organisms, which could have led to some of our our statistics not really lining up where we wanted them to. It is also possible that it could be performed again to collect more data points, not necessarily with the sites further apart, but more data points in general to at least further validate the significance of the results.